Thank you, Louise, and I'd like to thank the organizing committee and particularly Ron Mon for the opportunity to be here. I want to say at the outset, I am neither a nutritionist or someone that works with athletes, and so I'm incompetent to give this talk. But with that said, what I'd like to talk about is what I know about antioxidants and exercise. And my format's going to be a little different than some of the previous talks, and because of, I think, the difference in backgrounds of the individuals, I'm going to spend more time today talking about some of the background information of an overview between radicals, exercise, and antioxidants, and I hope that that'll foster discussion. My discussion that will be practically applied to a consensus statement, I'll try to address two primary questions. The first is, can dietary antioxidants improve exercise performance? And this will be primarily endurance performance because that's actually where most of the work has taken place. And then the second question is, do athletes require additional antioxidants above and beyond what you get in a normal, well-balanced diet? And then I'll try to come up with some recommendations. I'm not going to be so presumptuous to say that I have a consensus, but I'm going to make my recommendations based on what I believe the literature says. If you look at the field of free radical biology and skeletal muscle, it's actually a very new field. And in fact, the first paper describing that radicals were actually produced during exercise didn't appear until 1982. You can see from this bar chart of the annual publications of reactive oxygen species in skeletal muscle, most of what we know has appeared in the literature since 1995. And so it's a very, very young field in that regard. I want to point out that you all know that radicals are molecules that possess unpaired electrons and therefore tend to promote oxidation. But I want to point out as well that there's many non-radicals that are also oxidants that biologically are very important during exercise. And two key ones are hydrogen peroxide and peroxynitrite. Even though nitric oxide is a radical, having an unpaired electron, it's not very reactive. It has lots of biologically important roles, but it's probably this molecule that actually is more important in exercise and causing damage. And so it's important to appreciate that when we talk about radicals, we really sort of think about them as reactive oxygen species, an umbrella of reactive molecules that can cause oxidation. And the same is true for reactive nitrogen species. And the primary concern in an exercise situation is that when oxidants are produced in excess of the ability to scavenge them through antioxidants, the three primary targets in skeletal muscle that undergo damage are lipids that undergo a process called lipid peroxidation. There is some concern that under extreme conditions there may actually be DNA oxidation, but a primary concern is the oxidation of proteins. And collectively, this oxidative modification of these molecules have been postulated to cause both skeletal muscle atrophy and contractile dysfunction. I want to make a major point here is that when we think of radicals, we all think about them in a negative sense. But all cells produce oxidants, and they produce them for very important biological reasons. They play important roles in cell signaling. We know there's literally over 100 transcriptional activating factors that are redox sensitive in skeletal muscle. This is very important. In terms of both cardiac and skeletal muscle, we know that redox regulation controls calcium release, and that cardiac and skeletal muscle myocytes don't work well without oxidants. Later on, I think we're going to hear about white cell function and the role that oxidants play in normal white cell function. And of course, oxidants, nitric oxide plays a critical role in the regulation of blood pressure. So these aren't all damaging molecules. They're in cells for reasons. But what's important to appreciate that there has to be a balance. Redox balance is absolutely critical for normal cell function. And redox balance in a very simple sense is simply the balance between oxidants and antioxidants. If this balance is tipped toward the oxidant status, that is, the amount of oxidants outweigh the amount of antioxidants, oxidative stress occurs, and oxidative stress can be bad in extreme cases. On the other hand, too many reductants, too many antioxidants can result in an imbalance resulting in reductive stress. this too can be bad and can alter gene expression markedly. When you look at it from an applied perspective in athletics, if you look at the overall cellular redox balance, and that is the ratio of oxidants to antioxidants, if there is an absolute balance between the two, then there's no oxidative damage and we're in balance. If the oxidants outweigh the antioxidants and there is cell damage, oxidation to proteins, oxidation to lipids, but this can be repaired, then the oxidative stress occurs, but cells don't die. Under extreme conditions, when oxidants outweigh antioxidants, cell death can occur, and this is one of the triggering factors to apoptosis, and it's been postulated this may contribute to cell death, skeletal muscle cell death, and cardiac myocyte death. But from an applied perspective, this redox balance is determined by a wide variety of factors. Certainly the fitness status of the individual. Exercise training causes an increased expression of antioxidant enzymes in both heart and skeletal muscle. Genetics plays a role. Environmental factors. We're now beginning to appreciate that high temperatures cause increased radical production and contracting skeletal muscle at any given oxygen uptake. We know that high altitude or hypoxic environments also exacerbate the rate of radical production. Inhaled oxidants in a polluted environment also contributes to potential oxidative damage to lung tissue. We know that diet can influence the oxidative status. Exercise intensity certainly plays a role in that. We believe that in general the amount of radical production in skeletal muscle is directly proportional to the oxygen consumption of the muscle. Age may also play a role, as senescent mitochondria produce more radicals than mitochondria obtained from young animals. The gender issue is also of interest because of the potential role that estrogen may play as an antioxidant. And so there's some speculation that there's less oxidant damage in women, but this remains a really very controversial issue. If you look at the impact of the tissue antioxidant capacity and the level of damage at any given level of oxidant production, you can come up with a scenario that looks something like this. And this is just a hypothetical situation. What I've plotted here on the y-axis is levels of oxidative stress. Level 0 is no oxidative stress, a complete balance between oxidants and antioxidants. Level 1 is where there is an imbalance toward the oxidized side, but modified damaged proteins and lipids can be repaired. The cell recovers, and perhaps this plays a role in the adaptive process. Level 2, or the highest level, is where you actually overwhelm the antioxidant capacity and cells wind up dying. If you take 3 tissue hypothetical levels of antioxidants, low, moderate, and high, you can see that any given level of oxidant production, those tissues that have the highest antioxidant capacity undergo the least damage. But the point here is that only tissues that have really low antioxidant capacity wind up reaching the level where they're actually succumb to this pressure and wind up dying. So when we think about nutritional antioxidants, I'd like to talk a little bit about some of the common nutritional antioxidants, but more importantly, I'd like to make some general statements about what makes a good antioxidant, and I'd like to remind you about the importance of antioxidants working together in a network. You know, if you look at the popular literature, it's very common in every newspaper to read articles about vitamin E, C, certain carotenoids, with probably beta-carotene being the one receiving the most ink. But there are flavonoids and other plant phenols. There are over 5,000 of these compounds, that many of which have been shown to have antioxidant properties. You've read a great deal in the last several years about alpha-lipoic acid and its potential role as an antioxidant. And I want to remind you that cysteine is a very important amino acid that may play the rate-limiting step in the synthesis of a very critical, important cellular thiol, glutathione, that's very important in the overall antioxidant protection of cells. I also want to remind you that trace minerals such as zinc, magnesium, selenium, iron, all play important roles as cofactors for key endogenous antioxidant enzymes. But it's important to remember that not all antioxidants are created equal, and the structure is really the key component. The structure determines its reductant potential. The structure also determines where in the cell it's going to be located, whether it's in the lipid phase or the aqueous phase of the cell. When you think about nutritional antioxidants, it's important to consider both the half-life and the ability of the antioxidant to be recycled. When you put all this together and you look at the location perspective, it's important to think about that in order for an antioxidant to be to be successful, it has to be in the vicinity of where the oxidant is produced in order to scavenge this. And this raises the importance of having antioxidants in both the lipid phase and the aqueous phase of the cell. And I also want to remind you that this is one large network, that dietary antioxidants don't work in isolation from endogenous antioxidants, such as glutathione or antioxidant enzymes. And so, in order to maintain cellular redox balance, it is a cooperative interaction between dietary antioxidants and these endogenous molecules. Many of the studies that you've probably all read in the literature looking at the effects of nutritional antioxidants on performance have failed to recognize, in my view, the networking importance of antioxidant enzymes. And I show you this cartoon simply to remind you that these antioxidant enzymes, many of them are coupled in what we call redox cycles. And an illustration of this is vitamin E. When vitamin E accepts an electron and forms a vitamin E radical, it is no longer able to scavenge radicals. But it can be recycled by vitamin C, which then can be recycled by glutathione. Lipoic acid plays a role in this redox cycle as well. And you can see the interconnection between all of these nutritional antioxidants working as a network in order to regenerate each other. Let's talk a bit about antioxidants and the interaction with exercise. And even though the first paper that described radicals being produced in skeletal muscle was published in '82, it really wasn't to the early '90s that we actually appreciated the relationship between exercise intensity and the rate of oxidant production. And this is now a classic paper published in '92 by Mike Reed. And what he did in these experiments was load fibers with a fluorescent oxidant probe, dichlorofluorescein. And when it becomes oxidized, you can measure the fluorescent emission and quantify the amount of radicals being produced. And what Mike demonstrated in these experiments was that in relaxed muscle, there's oxidants being produced as well. But when the muscle's actively contracting, the amount of oxidant production goes up markedly. This amount of oxidant production increases in proportion to the amount of contraction, exercise intensity, but it is also amplified with the same amount of contractile activity in hot conditions when the muscle temperature goes up, and also under hypoxic conditions, which seems rather paradoxical given the fact that many of these reactive species are oxygen in origin. Where these radicals come from has been a topic of interest from a number of folks, and historically it was believed that they all came from mitochondrial sources. We now know that's not the case. There are many other oxidant-producing pathways in skeletal muscle. However, during exercise, it seems likely that mitochondrial production is indeed the primary source. And so therefore having antioxidants that are able to work in the mitochondrial membrane, the mitochondrial matrix, and in the aqueous area around the mitochondria seems very reasonable. Well, is there evidence that there's actually oxidative damage in skeletal muscle with heavy contractile activity? And the short answer to that is yes, and here's some evidence of that. This is some work from our laboratory in an animal model where this is an in situ preparation where the hind limb is stimulated to contract. This is a marker of lipid damage, lipid hydroperoxides. And you can clearly see in a muscle that underwent a 60-minute endurance protocol that there was almost a doubling of the amount of lipid hydroperoxides. This kind of study has been replicated all over the world in a variety of conditions in skeletal muscle. In terms of humans, we know the following. We know that human skeletal muscle produces radicals. We know that heavy bouts of exercise results in tissue depletion of antioxidants. We know that proteins are oxidized, just like in laboratory animal studies. We know that lipid peroxidation is increased. And there is some speculation that in intense exercise following marathons, there may actually be DNA damage in white blood cells, although this is a quite controversial area. Well, the fundamental questions I think that you're most interested in are the following. The first is, will dietary antioxidants improve athletic performance? The second being, does regular exercise increase the daily requirement of dietary antioxidants? Let's deal with the first issue first. What evidence is there that antioxidants will actually delay muscular fatigue? Well, in the animal literature, there's a lot of evidence. And this is one of the first studies, I think, that was very cleverly controlled that demonstrated this. This was published in 1990 by Shindo and colleagues. And the experimental paradigm here is an isolated diaphragm preparation where the animal was treated with a cysteine donor and a thiol compound N-acetylcysteine. N-acetylcysteine is thought to increase cellular levels of glutathione, and it also has antioxidant scavenging properties in itself. The muscle was stimulated to contract over a 20-minute period with rigorous contractions and allowed to recover for 20 minutes as well. And you can clearly see that in the muscle treated with N-acetylcysteine that the rate of fatigue development compared to control was markedly less. You can see that upon recovery, the amount of force production compared to the percent of initial force was better maintained. And this started a whole cascade of experiments using various animal preparations that demonstrated that in an isolated muscle, if you increase the antioxidant capacity, the muscle generally was able to generate a higher power output for a longer time period. Well, if you move to the human literature and simply survey the literature, and I'm not going to go through each of these studies individually, I'm going to give you my synopsis of what the studies say. There have been scores of trials over the last 20 years. These experiments have used both sedentary and trained subjects. They've used a variety of exercise modality, including running, cycling, or swimming. In some studies, blood and muscle chemistry has been monitored, but this has been rare. And in most studies, in almost all of these, performance has been the primary endpoint. If you look at these studies in general, many of them demonstrate that muscle antioxidants are protected by supplementation. There tends to be less blood glutathione oxidation after exercise. There is less blood protein oxidation. Lipid peroxidation, primarily in the blood, is also blunted, but there is no clear evidence of a performance benefit. So, given the fact that in animal models we can reduce oxidative stress by various antioxidant interventions, given the fact that many animal studies show that antioxidants can delay fatigue, you have to ask the question, why do these human studies not demonstrate a similar benefit? Well, I don't want to criticize all of the studies, but if you examine the literature, some of these studies are flawed that have been done. Many of them simply use one antioxidant as a nutritional intervention. Many of them use this antioxidant intervention for a very short time period. Vitamin E is a classic illustration. We know from the animal literature that it takes up to 14 weeks to significantly increase and reach a plateau in vitamin E levels in the sarcolemma of both cardiac and skeletal muscle myocytes. None of the human studies have approached this kind of nutritional intervention. Very few of them have used multiple networking antioxidants. So the design of many of these experiments is indeed in question. Some people have argued that perhaps these dietary antioxidants don't reach the site of oxidant production during exercise. I doubt this very much. I believe that it seems very likely to me that vitamin C and vitamin E both are in key positions that can scavenge antioxidants during exercise. Some people have argued that, well, a well-balanced human diet contains optimal antioxidants. Certainly a possibility. The other possibility, and the one that I subscribe to, is that it appears clear to me that antioxidant levels in cells are highly regulated. That if you increase the amount of dietary antioxidants to an exorbitant amount, you simply downregulate endogenous antioxidants to counterbalance that, to maintain redox balance. And that's the goal of the cell, to maintain redox balance. So the final question is, does regular exercise increase your need for dietary antioxidants? And in searching the literature, I think I would conclude a similar thing to Koenig in a paper in 2001. And his conclusion was as follows. Therefore, although the theoretical background may be sound, at present there is no scientific evidence to recommend that increased quantities of antioxidants is required for physically active people, exceeding the amount provided by healthy, balanced nutrition. I would like to also caution that the possibility exists, at least with megadosing of antioxidants, that you may actually impair muscular performance. And here's a study from our laboratory when we fed rats an extremely high diet of vitamin E and alpha-lipoic acid for a 14-week period. And notice the top graph. This is a force frequency curve. And again, this is in a laboratory animal, a rat, and this is a hind limb in situ preparation, where we stimulated the nerve at various stimulation frequencies and measured the force output. What you can see is that in the animals that received the high antioxidant diet, that this so-called force frequency curve is shifted down and to the right, indicating that at physiological stimulations of the muscle, there's actually inefficient excitation-contraction coupling, less force being generated, suggesting that you can have too much of a good thing. In another recent study from our laboratory, when animals were fed a rather kitchen sink diet, if you will, of antioxidants, a wide combination, when we exercise trained them, we found that those animals that were receiving the high antioxidant diet, that cardiac and skeletal muscle adaptation to this training was not comparable to the adaptation occurring in the animals eating the control diet. And in fact, for example, there was a suppression of exercise-induced expression of heat shock protein 72, 40, and 32 in response to exercise training, suggesting that high diets high in antioxidants can modify the way that skeletal muscle and heart adapts to endurance training. And that's led us to the conclusion that there's an optimal redox balance in skeletal muscle. That if you move too far to the reduced side, there's a problem. You move too far to the oxidized side, there's a problem. Indeed, optimal regulation is the goal. With that as a background, let me make some very conservative recommendations. And I don't call this consensus because the consensus will come from you. This is simply my recommendations based on the literature. It is my view that current evidence doesn't support the notion that dietary supplementation of antioxidants above that received in the diet will improve human athletic performance. I also think that there is currently no evidence that physical training requires antioxidant supplementation above the normal antioxidants obtained from a well-designed diet. I would caution athletes against the indiscriminate use, that is, megadoses, of antioxidant supplementation. Is there ever a case where you could recommend supplementation of antioxidants? I think possibly. I think that supplementation could play a role when food intake for some reason or another is restricted, or where there's a dietary deficiency of antioxidants that has been clinically determined. Thanks very much.